ideas, but yeah, one thing is to look at acrylamide gene interactions, which I am now doing in my postdoc project. We got a funding from the Dutch Cancer Society, KWF, to look within the Netherlands cohort study for specifically hormonal cancers, if there's an interaction with certain genes. So do certain genes lead to higher risks than, I mean the variety in those genes, does it lead to a higher risk than other varieties of the gene? And we will be looking at single nucleotide polymorphisms in acrylamide metabolizing enzymes, but also in sex hormone receptors and sex hormone metabolism, because of this hormonal hypothesis that we have. And if, for instance, it turns out that people that have an efficient estrogen receptor have higher acrylamide-related risks than people with a less efficient receptor, that also tells you something about the mechanism, right? It tells you that it may be indeed a hormonal effect going on, and that would give credibility to the association that you saw. So the acrylamide leading to higher ovarian endometrial cancer, which are hormonally, arise hormonally. So that's one thing. Another thing is an in vitro study with human cells. It's called CALUX. I said here that it's in progress, but actually I heard the results yesterday evening. I'll tell you about it. It's human cells, breast cancer cells, for instance, and it measures if the acrylamide or glycinamide that you add to the system, they bind and activate hormone receptors. So there are several receptors that you can test: estrogen, androgen, progesterone. And the principle is that the compound binds to the receptor, activates it, and then the complex, so the ligand and the receptor, they move to DNA, bind a responsive element there, which leads to transcription of genes. And normally those genes are under the control of the estrogen receptor, but now they have built in a gene that, when activated, gives light. So you can measure this light, and then the intensity of the light is a measure of how much the estrogen receptor is bound and activated by the compound. But yesterday I heard acrylamide and glycinamide do not bind the receptors. So at least we know now that it is not, if acrylamide works in a hormonal fashion, it is not through binding directly to the hormone receptors. There must be maybe other ways that it does so, like influencing the metabolism of sex hormones. And then there's an intervention study I would really like to do, and of course it's very ethically a problem, because you expose people to acrylamide. That's the problem. That's also the reason why we do animal tests, because we cannot test directly on humans. But this has a bit of a different setup that I hope that the ethical committee will approve. I'll tell you. So we are writing a grant application for this, for the World Cancer Research Fund, and it's a collaboration with the Department of Toxicoeconomics I just told you about. And it will involve 70 postmenopausal women, because those are the women that we saw that got the endometrial ovarian cancer, all non-smokers. And we will give them 50 grams of potato crisps. And maybe you as a student thinks, well, 50 grams, it's nothing. I eat that every hour. No, but this is, so we thought if we give them crisps with a low acrylamide intake and crisps with high intake. So the only thing we hope will differ between the groups is the acrylamide level, because the fat and the salt intake of the chips we will keep comparable. But we hope that the medical ethical committee will approve this, because we really have to make a point that we really think this is necessary to do this, but also because, yeah, you can just buy crisps in the supermarket. I mean, it's not unethical to do this, because people do it to themselves every day. And also, this 50 grams is determined it's a high normal intake, so there are a lot of people actually eating this amount of crisps. So, but it is to be determined if the medical ethical committee will approve this, we hope. And of course, we do not expect, we will give them it for 6 weeks, and we do not expect a 6-week period to have any detrimental health effects at all. And what we will assess in these people is changes in sex hormones before and after. Also micronuclei, which are a validated marker for carcinogenesis, marker for genetic damage. And also gene expression changes. Because, yeah, gene expression changes really show the first molecular events happening after an exposure. So they may give indications for what goes on, which genes are affected, and do they fit in certain pathways, like for instance a genotoxic pathway or a hormonal pathway, or maybe even other pathways. So that's yeah, that gives really interesting data, we hope. But so this is not happening yet. Oh yeah, I was going to tell you. Oh no, I didn't tell you. Yeah, that unfortunately, yeah, it's always a disappointment as a scientist when your hypothesis is not. You do not find support for your hypothesis. But yeah, we will move on. So about this, these molecular studies on acrylamide and cancer, I could imagine that maybe some of you find this an interesting topic, and if so, if you're looking for an internship together with the Department of Toxicogenomics, or even for a PhD project after your master's, please contact me and find out if there is something to do at that time. But not if you're looking for a low-profile topic, so expect that your research will get attention, and you have to be prepared to talk to the media and yeah, take responsibilities for your findings. So that's it. So thank you for your attention. I would like to thank my partners in troublemaking from the Department of Epidemiology, and also Tino and the Voedsel- en Warenautoriteit. And yeah, I'll now take questions if you have. Was it clear? Was it clear? Yeah.
Well, I have a question about the intervention study you mentioned at the later slides. You just, well, it's a kind of application of the ethics commission, and I was wondering what the participants in the study of the 70 post-menopause women are given on information, because I can imagine if they found out they worked in a study which is related in one way to cancer, I would find some mess.
Yeah, that's a good question. I think yeah, you always have to give an extensive information letter to the participants to get their informed consent, so they know exactly what is going on, that why we are studying it, and that they can always withdraw if they want. Things like that have to be in that letter. But indeed also about why we study it. And I think that will be a debate with the medical ethical committee. Exactly how much information do we give? Because we study it because it is not really yet known if there is danger in humans, right? That's what we want to find out. But our hypothesis is that it causes sex hormone changes. And well, yeah, this DNA damage, of course, we also measure, but yeah. DNA damage is also a transient thing. It may only matter if you are exposed for a very long time or your whole lifetime. So that will be indeed a debate with the medical ethical committee to see what we have to, what they want us to put in there. Yeah. Yes?
So again, at the intervention study, will all participants get the same diet for 6 weeks?
Ah, yeah, that's a good question. It's also something that yeah, we have to think about, of course, and also what the medical ethical committee will want to hear, because they also want to make sure not only that it is ethically responsible, but also that you perform a good study. Because if you do not perform a good study, it's unethical anyway. Yeah, that you experiment on people with a very bad design. So you have to. But yeah, no, I don't think we will give a specific diet to them, provide them with diet. We might ask them to keep dietary records for a few days to see afterwards if their diet intake was comparable. But this would be relevant if you would expect their diet to change and to differ between the two groups. But they both get chips in the same amount. So,
for example, if someone eats a lot of meat or roasted meat, the acrylamide intake will be higher.
Oh well, not in meat. Meat is not very important source for acrylamide, but yeah, I get your point. Yeah. So yeah, if you eat a lot of meat, indeed, but the study groups are of such a large sample size that, so we at random determine who gets the high acrylamide chips and the low acrylamide chips, and you would not expect to be, if you randomize it, to be different, the dietary patterns of the people, right? So you would expect to see as many high meat eaters as in one group as in the other. So that would cancel out. That's what you think, but indeed by those dietary records we can have some clue about that. To see if it is indeed was randomly distributed among the groups. But to give a diet, it's much more expensive. It also still does not guarantee that people actually eat this diet and they don't eat other things besides that. And we wonder if it's really necessary to do that. But it's something that we will indeed have to think about. Yeah. Other things?
How can you measure acrylamide?
So acrylamide in the blood. So you mean the adducts to acrylamide? I think you first isolate the red blood cells. Of course, you lyse them, and then you split the adduct off from the hemoglobin, and then I'm not a technician on this, but I think it's determined with GCMS, the specific fragment, this adduct, and then you spike it with a concentration of this adduct to find out how much was in the sample. But it's very expensive. It's very expensive measurement, so that is a drawback for just every study using these biomarkers. I mean, it's very expensive. So if you want to study a real good large group, which is also important, the drawback is that those biomarkers are so expensive, and your study may not be feasible anymore in a financial way.
And the study you want to measure before and after?
Yeah.
What
Yeah, could also be. Yeah, but I'm already having problems staying within the budget. So it's always, you always have to make compromises somewhere. The idea is to get it past the jury of the grant committee and the medical ethical committee at the same time. Yeah. Yeah, I will. Yeah, that's a matter of indeed external validity. So, are your results representative of the whole population? Because always you study a part of the population indeed, and can you extrapolate it? Are the results also valid for other parts of the population? And I think it is because we try to have as the only difference between the groups. They probably will all be from the area around the university, because I will put advertisements in local newspapers, for instance, to attract women to come. But we hope that they may have a very comparable diet containing acrylamide, and other populations may have very other sources contributing to their acrylamide intake. But in this case, we hope that the only difference between the two groups is the acrylamide intake itself. So that's, I hope, if we see something, then we can attribute this to acrylamide itself. So we do think we can use this conclusion to say, in general, also in other populations, there would be a risk related or effects happening due to acrylamide exposure. Yeah, that is very likely, because for instance, Asian populations, Afro-Americans are known to have very different occurrence of genotype variants in genes in metabolism. also in those acrylamide metabolizing genes, so that could well be have an effect. But also, if we have enough money, I would like to look at that. Although our group is very small to really look at that, but we would like to measure genotypes, so polymorphisms in genes also, and if they have a different effect on what we see. So, but I don't think we will be able to do it. Maybe afterwards we keep the blood samples, and we can, if we find additional funding, we might always do that afterwards. So, yeah. Okay, that's it. You will have another lecture right after this. Okay, but maybe you have a few minutes for coffee, if you're still there. Okay, thanks.